So it's just kind of interesting because I feel like this is a new experience for me, and one of the things that I wanted to do was also give an introduction to my organism of choice. I'm going to turn things down just a tad more because I've got some. Yeah, there we go. So I work on Myxococcus xanthus, and Myxococcus xanthus is a little different than the last bacteria we just talked about because. Well, for those of you in the audience that care, it's a gram-negative delta proteobacteria, which tells you something about sort of its structure. And, for example, Myxococcus xanthus, I think, actually stands for gold slime rod. And so you can see that it's rod-shaped. Let's see if I can actually make that do that again. It moves by a form of motility called gliding motility. Now, there's some controversy about exactly what that means, but for the point of this talk, it doesn't really matter. Suffice to say, it can't swim. What it can do is that it moves along a semi-solid surface like an agar surface, and it moves along its leading edge or its leading pole, and then it can reverse direction occasionally, which you were watching it do there. And then the last thing is that it doesn't exist in the form you just saw it in as sort of individual cells. That doesn't really occur in nature. It exists almost entirely in isogenic biofilms that are called swarms. And I'm hoping that's not good. I don't want to shut down. I'll log out.
Are you saying other universities have better projection than Syracuse?
All right. What I'm going to do is I'm going to keep talking, actually, and we're going to pretend like I'm giving this talk while. All right. So okay. So like I said, Myxococcus xanthus exists in isogenic biofilm called the swarm. And the thing that Myxococcus xanthus does that sort of puts it on the map in terms of self-organizing systems is that a biofilm of Myxococcus or a swarm of Myxococcus xanthus, when you start it, will self-organize into sort of mushroom-shaped structures that are called fruiting bodies. Each fruiting body contains 100,000 cells, approximately, that were once sort of relatively evenly dispersed throughout a swarm. I'm kind of terrified right now. I really don't want to run FSCK, but I think I'll have to. And while my computer is vomiting, I'm going to keep on going, and we'll just pick up where we have to. Yes, please fix everything. I don't want to give you my root password. What I want to do is just have you restart as you want. And this is going to truly be a sure moment. I have it on an SD card, but somebody would have to have open office. All right, if this asks me for my root password again, we'll do that.
Like Bill Gates is winning, only Linux and Mac have given problems so far.
Bill Gates has infiltrated these things.
All right. plug in. Same thing. It's peripheral. That's the last time I go back and run a movie over here. Okay. So here is the sort of quintessential introductory slide to a Myxococcus xanthus talk. The myxobacteria, this represents a schematic of the xanthus life cycle. So when there is sufficient food, they grow what is called vegetatively. Here's a microcolony that is swarming on nutrient agar. If you introduce nutrient stress in the form of starvation, it will go through a series of patterns, which we'll get into more detail in just a second, but the sort of most pronounced of which is the formation of traveling waves. These are actually non-Zhabotinsky waves. And following the formation of waves, they will begin to form these aggregates. Now, these are actually scanning electron micrographs of M. xanthus swarms forming one of these aggregates. So the aggregates continue to grow and begin to rise off the surface of the agar plate and eventually form a mature fruiting body. This is actually almost visible to the naked eye. It's about 1/10 of a millimeter across. So inside the fruiting body, the cells on the interior of it will actually change shape and become rounded and both metabolically quiescent and environmentally resistant spores. And you can actually see them because if you freeze fracture a fruiting body, you can actually see the myxospores on the inside of it. But that's not the only pattern. So, okay, the reason why development is the predominant pattern that is studied in Myxococcus xanthus is that it has a verifiable endpoint. If at the end of an experiment you have visible fruiting bodies and you test them for environmentally resistant myxospores and it has myxospores, you know you've successfully undergone development, it's quantifiable, it's verifiable, but it's definitely not the only pattern that Myxococcus xanthus exhibits. So this is actually Myxococcus xanthus swarming in time lapse on minimal nutrient agar. And this is about 3 million cells in an area of about 1 millimeter across, I'd say total. You can actually see individual cells or nearly individual cells. These larger groups are clumps of either hundreds or thousands of cells that are sort of moving. I wouldn't want to say as slugs, but they're sort of moving in unison. And as these things are moving across the agar, they're leaving behind this slime, which seems to be some sort of signaling that they do because they predominantly follow slime. And if you were watching sort of different parts of this movie, you'd actually probably see a whole slew of different patterns. So, for example, you'd see swirls of cells that are moving around, or you'd see these beginning aggregates that will eventually become fruiting bodies. At least a subset of them will. You see that they actually do form sort of discrete tiers of cells. There was actually a paper on that by Larry Shimkins not too long ago. And then last, you see these sort of rafts or slugs. These are hundreds or even thousands of cells that are sort of moving in unison. So development and the formation of fruiting bodies is not the only pattern that Myxococcus xanthus exhibits a whole slew of patterns sort of on the way to it. Now, okay, this is where I come at it from, because I started out as a biochemist and molecular biologist that was interested in trying to figure out how you can get things as simple as the instructions that genes can process to make three-dimensional structures. And that was part of the reason I got interested in Myxococcus xanthus, because Myxococcus xanthus makes some pretty simple three-dimensional structures, and at the time I was choosing a model organism, and we seemed kind of far away from it anyway, so I thought, why choose something complicated like C. elegans? I'd choose something a lot simpler. And in 2005, we actually got the genome sequenced, and it turns out that Myxococcus xanthus has a single circular chromosome with 9 million base pairs. And in those 9 million base pairs of DNA, there are approximately 7,500 open reading frames. Each open reading frame is either a gene which encodes for a protein, or at least something that looks an awful lot like a gene that encodes for a protein, but not all of them do. So we call them open reading frames, but you can assume that probably 95% of them will wind up encoding for proteins, so assume that there's about 7,500 proteins. And the function of about 25 of them is completely unknown. So here's actually the map, and it's color-coded, so this is actually the role of the genes. They're sort of clustered together according to functional roles. So you've got things like cell envelope and fatty acid and phospholipid metabolism, biosynthesis, and you get down here to things that are a little bit more or a little less clear. Protein fate, cellular processes. So when I say that the function of approximately 25 is completely unknown basically means unknown function here, and conserved hypotheticals, which basically means unknown function, but there's proteins of unknown function that are the same in other organisms that have been sequenced. So when you add that up, that's 25% where we either don't know what it is at all, or we don't know what it is, but there's something else in another organism that we don't know what it is. But then when you look at some of these others, like cellular processes, okay, so 5.5% cellular processes, but that doesn't really tell you very much about what the protein does, really, and it doesn't tell you anything about what the protein does in light of how Myxococcus xanthus functions in relation to its environment and each other. And so this is actually a slide that seems to be sort of in a lot of the talks, since this is about emergent behavior, which is that what we've got essentially are sort of three levels, or three sort of scales of complexity. So you've got gene function, you've got individual genes, or 7,500 individual genes. Each one does essentially one thing. Genes are relatively simple machines. It's not like there's a lot of variability once we've made it. You then have cell movement, so you've got the individual cells. We saw a movie of those. They go back and forth along their long axis. They occasionally reverse direction. They're semi-rigid rods, and they only move in 2 dimensions. They can't swim. And then on the last, you have global self-organization, which is the idea that you can actually make patterns if you put millions of these things together and they're signaling to each other and moving in unison. And the problem is that there's actually a barrier here, because each one of these scales is sort of, at this point, impossible or nearly impossible to sort of move past. So it's right now we're working on it, but we haven't figured out exactly how cell movement leads to global self-organization in Myxococcus xanthus, or for that matter, exactly how genes function together to control cell movement. And here's sort of the point of the talk, at least, and the point of what my lab is trying to do right now. It's not like it's completely hopeless. We've got some things that we can actually use to make some predictions and maybe make some good guesses, especially about the 25% of proteins we don't know what they do. So, for example, between gene function and cell movement, well, you've got functional limitations to proteins. Proteins are infinitely complex. They do essentially one thing and one thing only, so you've got 7,500 instructions that you can sort of put together in some meaningful way to include all of sort of cellular structure and metabolism, as well as things like motility in general, not to mention cooperative motility. And then you've also got the fact that, at least in this case, you're dealing with prokaryotic genetics. So all of the sort of organelles and vacuoles and all the sort of complex stuff like that you have with the eukaryotic cell, you don't have here. It's not a drastic oversimplification to say that a bacteria is mostly a bag of water. So you don't have as much sort of structural stuff that you have to worry about, like local concentration gradients and diffusions across organelles. So you've got that simplifies this connection between the cell movement and global self-organization. Well, you're dealing with an isogenic population. Granted, it may not be exactly the same. There may be differences in gene expression, say, in the middle of the swarm versus at the edge of the swarm. But you definitely have it. It's definitely isogenic, and it's probably more homogeneous than most self-organizing biological systems. And then also you have movement limitations. So the thing doesn't swim. It moves in two dimensions. It only moves along this long axis. They're all rods. It's not like the cells take on different shapes. So you've got that. And then between gene function and global self-organization, you've got clustering based on gene homology. So you might not know what a gene does in a particular organism, but you can start clustering them according to sort of what they do in other organisms, or what other genes that look like it in that genome do. That didn't really make a lot of sense. Then you've got also the fact that you know it's an emergent property. I mean, you've got the patterns that are leading up to the transition from a less ordered to a more ordered state. And there are certain things that you know about emergent properties that you can guess, like they're based on multiple interactions between relatively simple instruction sets and all that good stuff. That's actually probably one thing I don't have to talk about too much to this crowd. And so, all right, that said, here's the problem that I was facing when I sort of started attacking this problem in this organism, is that we've got sort of a really limited set of tools that we can use to get any kind of quantifiable metrics. So we can measure growth rate. Like if we make a mutant in a gene, if we make a mutation in a gene, get that strain growing up, does it grow any slower? Or if we put it on agar, does the swarm expand, nutrient agar, does the swarm expand any slower than normal? We can also cripple different motility systems. It turns out Myxococcus xanthus has two genetically separable motility systems. We can cripple one or the other and see if maybe that changes the rate of swarm expansion. Then also the rate of aggregation. Does it make fruiting bodies at a normal time? Or is it delayed? And then lastly, does it make spores? So we can, again, see at the end of development, we can stress the system by heating it and then seeing whether or not the spores were made, because if they weren't made, then all the cells will die, and if they are spores, then they'll be heat resistant and they'll survive. And so this is pretty much what we've got to test mutants. So based on these criteria, it's been estimated the majority, somewhere around 90% of genes, are going to show no phenotype if you knock them out and then grow up the organism. They'll still do all of this stuff within some sort of statistically significant, sort of near wild type level. So, all right. To all the other molecular biologists in the room, I apologize. This is a drastic oversimplification, but it gets the point across. So what we've done now is start taking a look, rather than individual genes that exhibit interesting phenotypes and focusing on those, to start focusing on the larger groups of homologous genes. So these are three types of genes. All of them are related to signaling. All of them might have something to do with self-organization and in Myxococcus xanthus. One is a group called the NTRC-like regulators, which are part of two component systems. And it turns out that Myxococcus xanthus has a total of 53 of these in its genome. And so if you've got a signal, this little orange triangle right here, it gets sensed by one protein, which passes a phosphate to another protein, which is bound to the DNA, influences transcription somehow and makes a new product, and that influences how the cells respond to that input. You then have another type called an ECF sigma factor. These are also known as these are part of one-component systems. It turns out that Myxococcus xanthus has a total of 28 of those. And in this case, it's a little bit different. In this case, the signal binds to the protein. That protein itself, rather than passing a phosphate to another protein, binds to and influences the transcription of DNA, puts out another set of new signals, and can alter the cell's behavior. And then the last one that I'll bring up are the ABC transporters. These are importers or exporters. They can do either one. And all right, so they're not actually sensing and responding to the signal. Instead, they're sort of gated channels that could let a signal in. Once that signal's in, who knows what it could do, influence the transcription of another gene, which would produce another product, which actually might get exported by another ABC transporter. So each one of these kinds of genes could be, or proteins could be involved in self-organization. And it turns out there are 120 ABC transporter systems in Myxococcus xanthus. I think if you add up the genes, and sometimes there's more than one ORF related to them, it's like 198 ORFs that are in there. So the reason why I bring these up is each one of these homologous families were found because one or a couple of these produced an interesting phenotype. So a researcher, either me in the case of the ABC transporters or somebody else, actually went ahead and disrupted the rest of them. All 53 NTRC-like regulators are all ECF sigma factors, and the vast majority of them produced no phenotype that is different from wild type in any but that one that was discovered to begin with. So, all right. What we decided to do was to start trying to see if we could find new metrics that we could use to differentiate between the phenotypes of these genes that right now present us with no aberrant phenotype. So before I get on to mutants, this is actually going to be development of DK1622 as wild type M. xanthus, but I think on the slides I refer to it as DK1622. This again is time lapse. It's sped up, so I think every second you're watching this is about 15 minutes in real time. This is about 6 million M. xanthus cells on starvation agar. And it doesn't take very long for them to pick points in two-dimensional space, form aggregates, and then those aggregates eventually stabilize into fruiting bodies. Now, if we break those up into 4 frames, what we notice is this. It starts out essentially as a mottled bunch of gray. Relatively quickly, there are, within a few hours, these points of aggregation start, and they grow, but then at some point, they go from lots here to fewer right here. And that was actually the point that we wanted to focus on. We actually didn't realize that at some point we were going to lose aggregates. And it turns out if you watch the movie again, you can see when that happens. First, a lot of aggregates form, and then about half of them actually, it turns out, either move and merge into other aggregates, or they just dissolve. And when we plotted that, well, before we did that, so we started looking at the ECO sigma factors, because they were actually the most normal looking. There were 20 out of the 28. One gave us a phenotype that was a developmental defect. The other 27 looked pretty much like wild type. And so we started doing that with the ECO sigma factors. This is actually a mutation in the gene MXAN2405. If they just start at the top, at the origin of replication, and just number them from 1 to 7,500, so the number doesn't actually have any meaning. We made the same kind of movie here, and it goes ahead and in a reasonably timely fashion, it makes aggregates, and those aggregates actually appeared to move a little bit more than they should have, but eventually stabilize and form fruiting bodies. However, if you take a plot, and on the x-axis this is time in hours, and on the y-axis this is actually the number of aggregates per unit area. Here's wild type. First you see a lot of aggregates, and then it plateaus, and then over a relatively short period of time you lose about half of them, and then it stabilizes. Here's your mutant. So here instead you get a slower increase, but you actually get more aggregates, and then you don't get that. They're sort of merging, and so a few disappear over time, but you don't get this period here where you get this decrease where they're merging and disappearing. And if you look instead of at aggregate number, if you look at aggregate size, here's again development in hours, and here's the average size of aggregates, and you get this little dip here, right, where that you lost a bunch of them, because if you notice the ones that were sort of shrinking and dispersing, first they would shrink and get smaller. So this is a little transient dip in the average size of a fruiting body, and that isn't shown at all in MXAN2405. So, all right, that's a relatively subtle but quantifiable phenotype, but there's another sort of interesting element to it. In collaboration actually with the lab of Oleg Goshen, Oleg is developing an agent-based model based on the best that we can do in terms of a model for how aggregation occurs, which is something called the traffic jam model, which basically says that there are predetermined points based on sort of collisions at odd angles where aggregates start. Those aggregates essentially are where cells pause. They accumulate as other cells collide with the with the cells that have already paused. Here's a simulation of it. And so the aggregates do form and grow and eventually stabilize. And when we take a look here at aggregate number with time versus the number of aggregates, here's wild type, and I shaved off the last little bit here in terms of time. So here is the dip in aggregate number for wild type, and then the dotted line here is the mutant MXAN2405, and then there's the traffic jam agent-based model is the hatched line. And if you look at aggregate size, again here's wild type with that dip, and here is the mutant 2405, and then here is the traffic jam model. That just keeps on going up. So, all right, and here's the last thing. And again, I'll probably do a poor job of explaining this. I'll apologize to Oleg to begin with. The last thing that we wanted to do is try to figure out whether or not there was any order in the placement of fruiting bodies. Because again, we were looking for metrics that would be able to differentiate between wild type and mutant. And so what Oleg came up with was actually using something that he called the cumulative radial distribution function. So just to make a long, for a biologist, a long equation short, essentially you're just looking at it's a function that looks at the number of aggregates in a circle as the circle increases, and then it generates a ratio, which is the frequency in that radius of whatever that size is over the frequency over an infinite area. And so essentially, as this function approaches, as R approaches infinity, it'll go from 0 when R is 0 to 1 when R approaches infinity. So basically, if the distribution of aggregates is random, it will approach 1 more quickly. If the distribution of aggregates is not random, it will approach 1 more slowly. Here is the plot of the radial distribution function for a bunch of different things, and I'll go through them one at a time. So first of all, the red line is a randomized control. So that's where the location of aggregates was just randomized so that we could get the radial distribution function for that. Then the purple line here is actually the agent-based model. So there's the traffic jam-based agent-based model. But then here's what's kind of interesting. Here's it actually turns out that the mutant actually isn't on here. That's too bad. The mutant actually is up here as well, but that the DK1622, so wild type in phase 3. Now we numbered those phases, and the phase in which you lost a lot of fruiting bodies was actually what was phase 4. So phase 3 was when you had a lot of aggregates before you lost about half of them. That actually is right up there with a random distribution, and by the time you hit the final distribution after you've lost about half, it actually turns out that those aren't random anymore. And so that's another weird difference. So in addition to the mutant not losing half of the fruiting or half of the aggregates halfway through development, it also seems that wild type, when it's losing half of its aggregates, is going from a more random to a less random distribution of aggregates. So just to summarize that, so DK1622, I forgot to number them, it has 5 distinct phases. During this 4th phase, a developing swarm of wild type loses approximately half of its aggregates, and the final arrangement of aggregates is ordered within a swarm. The arrangement of aggregates is not ordered prior to the 4th phase, and both the 4th phase and ordered aggregates are lacking in MXAN 2405 and the traffic jam agent-based model. All right, so I want to show you 2 more mutants. These are both ECF sigma factors as well. And what you'll see is that it's pretty obvious they look different from wild type. So here's one, MXAN 3686. And here's the other, MXAN 4773. Now again, really different from wild type, but at the end of the day, you've got aggregates that have spores in them. As far as a mixobiologist is concerned, they're wild type, even though they're clearly different in the way that they make aggregates. So, and this is very new data. We're still sort of developing. This is actually the work of a brand new graduate student in my lab. He's actually sitting in the back, I feel. Where we were trying to figure out, again, another metric where we could differentiate from mutants from wild types. So Phil is actually an expert in image processing. So he took, and I told him that you didn't have to know anything about xanthus, just take a look at these movies and see if you could come up with features that would differentiate one set from the other. So here's actually a piece of one movie, and what he did is he measured the change in pixel intensity for each pixel for frames n-1 and n, each frame in a movie of, let's say, 600 frames. Then he eliminated, so what that does is, and then he color-coded one red and one green. Now, of course, everywhere where you lose in pixel intensity, you're going to gain it somewhere else, so it actually sort of averages that to 0. So instead, he eliminated all the pixels that showed a decrease in pixel intensity, and then he plotted over time the sum of increasing pixels, both in number and intensity, for wild type and mutants. So this is just an example of wild types that are DK1622. This is the total upward intensity increase of sort of an entire frame or a set of frames, and this is over a 24-hour of development. So this is actually in frames, the x-axis here. And here's what's interesting. So we did this for two sets of, two independent wild types, and then for both of those mutants you just saw. And so here's wild type A and B, so those are two independent repeats, and then here's one mutant and then another, and then here's, again, the other mutant and again. So it looks like this, at least we're on to something where, again, this would be another way of looking at two movies of two different developing strains and seeing whether or not we can differentiate between the two of them. So, all right, where am I going with this? Because that doesn't really make a lot of sense to some of my colleagues. So we're going to continue to make insertion disruption mutants for sets of ors. Not just sets of homologous families that may be involved in signaling, but especially also the hypotheticals. But we need a set that's big enough so that we can start clustering them according to these features that we're developing. Then we're going to develop new features and characterize these mutant strains at swarm scale. So the kind of stuff we're showing here, I'm hoping to actually maybe come up with, if not dozens, hundreds of metrics that we can use to analyze these things and start differentiating between them. Then we're going to actually start grouping the open reading frames, the mutants, by clustering them according to feature characterization. So this sort of comes at it the way that I saw people dealing with microarray data. So we just went through sort of a lost half-decade where every molecular biologist was trying to cluster microarray data. But the clustering was actually interesting because it didn't have to be just expression data. You can cluster just about anything. So what essentially we're going to do is develop a feature set for each one of these mutant strains and then cluster the mutants that we characterize based on this set of features. And then lastly, to start examining, when we have a cluster of genes that all seem to behave the same way at a global scale, then we'll go in with fluorescently tagged cells and we'll start examining the behavior of individual cells and then incorporating that behavior into the agent-based model to see if we can't get closer and closer to a point where the agent-based model reflects what we're actually looking at a global scale. And then the obligatory acknowledgement slide. This work is done. Oleg and I have been collaborating since Oleg was a graduate student and I was a postdoc. And so I guess, Oleg does the theory, I do the experiments, but I can't really tell you which one of the ideas is mine and which one's Oleg's because we're in communication so much. So that just said. And then Qingyuan Yan and Claire, actually, that's what I should first be called, are both graduate students in my lab. Qingyuan is doing most of the mutant generation, and Claire is actually our new microscope expert. And Phil was the one who did the image analysis, the second one that I showed you. And then Stuart Angus actually got started on a lot of this stuff. He was an undergraduate here. He's now a PhD student at the University of Chicago. And Brian Mansfield actually just graduated this fall and is now a PhD student at Pittsburgh. And I would also like to thank NSF for funding this through a career award. But I always forget to thank them. That's a bad thing to do. But thank you very much. You're tough.
Well, this is, those pictures, I'm very excited, looking at those clustering pictures because I work on similar issues on clustering in glioma cells. And my question is the following.
What kind of cells?
Glioma, cancer
okay.
glioma cells. So in physics and material science, there is a process called coarsening, where you start with a homogeneous system, and then due to phase separation, clusters appear, and the process is the following. Small clusters lose cells, and those cells attach to larger clusters. So larger clusters grow at the expense of smaller ones. Now, in this situation, the number of clusters decreases all the time, and the average size of a cluster of clusters increases. Now, in your case, I see that the system freezes at some point, so the cluster size distribution function doesn't change anymore. So, do you have some intuitive explanations of what happens? Why cells stop proliferating and moving?
You're talking about Oswald, right?
Yes.
So I actually, Stuart started using terms like that, and I called up Oleg with that idea, and Oleg basically said that I needed to do more reading and I shouldn't use those terms until I really know what they mean. Yes, that's our guess as to what's happening, and actually Claire's project right now is to tag. So we have a system where we can tag cells fluorescently. I actually think I decided not to use one of these. Hang on. We also get Lee's gangrines, where we can tag cells fluorescently. And there it is. No, that's not it. Here we go. Tag cells fluorescently and then track them. And these are actually, that's the aggregate that's growing, and these are individual cells that are moving toward it. And so we're now going to repeat this experiment for aggregates that are increasing in size and aggregates that are diminishing, and seeing whether or not we can see a difference in the number of cells that are leaving the aggregates that are diminishing, because that would actually be a strong indication of coarsening, right?
Yeah, I mean, this is fantastic. I just don't understand why this freezes at the end. So why cells stop moving at some point?
Yeah, I mean, at some point, like, you ever, this isn't it's not just a physical phenomenon. There's a genetic element to it as well. So there's probably some genetic signal that turns on and tells them all, like, enough's enough.
Okay, I have a couple of quick questions. The first is about, you kind of mentioned that the traffic jam model is based on the assumption that collisions cause kind of local pauses and then aggregates form around that, which makes me wonder if you have or if you've thought about putting in some kind of little posts that act as obstacles to try to nucleate aggregates in specific areas.
So that's actually one of the things that makes working with Myxo. Myxo is, for those of us that are on a first name basis with it, that's what we call it. So Myxo will make a fruiting body on almost anything. So a glass bead, a little piece of dirt, you hit your pipette tip on the auger, it'll make a fruiting body right there. So we actually go to Herculean efforts to try to avoid anything like that. But yes, if there's an object that sort of gets in its way, it tends to sort of cluster around it, and it will actually make a fruiting body there.
But that means that you can have a possibility of, say, making an ordered array of where the fruiting bodies would appear and then seeing what that does for the interactions of the aggregates.
Yeah, it might be a really interesting idea.
And then just another quick question. One of the big differences, it seemed like, in the difference between the wild type and aggregates is that the wild type actually takes a lot longer to start forming aggregates initially, and I was wondering if you could comment on that.
So we've gotten variations in both directions. So the ones that we showed you, I guess, look like they formed more quickly. Actually, the last two mutants that I showed you, the two that I sort of showed you one after the other, those I don't know if they were started right at t equals. So you set up these slides, and you basically have to clamp them and incubate them, and it might sometimes take some time to sort of get that started. And then you've got to get the microscope started, so there's some time variation in there. So by the time you're ready to do a slide that actually is a solid experiment, like you have that all set up, but those two might have been started like an hour late.
So what's t equals 0 isn't necessarily well defined.
It's well defined when we're actually getting publishable data.
I have two questions. Do you know what is the mechanism why they aggregate? Is it like, access or something?
Well, so it's kind of an interesting example of well, in my opinion, of convergent evolution between this and another much more complicated organism called Dictyostelium discoideum, because they look really similar. I mean, Dictyostelium discoideum is amoeba, it's a slime mold, and it does many of the same things. It forms aggregates, it forms fruiting bodies with these spores, and it makes, so. But in the case of Dictyostelium, it is chemotaxis. But in Myxococcus xanthus, it appears not to be. In fact, they've spent a couple of decades trying to find chemotactic signals, and no one has been able to identify a fusible signal that Myxo will chemotax toward as an individual cell. That said, it doesn't. Sort of not identifying something is never proof that it doesn't exist. But in biology, it's really hard to prove that something doesn't exist.
We should talk, because I think somebody came to my lab with the same organism asking me to test for chemotaxis for one of the chemicals, but I don't remember what it is.
So it will chemotax as a swarm to certain things,
right.
but individual cells don't seem to be able to do that.
Well, we should talk. So that's not the case in this experiment we did in my lab. It's very short, but we will talk.
Just very quickly, some groups of Myxococcus feel that an alteration in the frequency of reversal constitutes chemotaxis. But one of the definitions of chemotaxis is that the cell actually gets somewhere. And so simply changing or going from this to this is not chemotaxis because you're not actually getting anywhere. So I don't know whether that's the experiment you're talking about, but a change in reversal frequency is not something that I define as chemotaxis.
The other question is, does the aggregate always happen in any kind of alga, or do you have to manipulate the mechanical stiffness of the alga, or humidity, or what is it easy to get aggregates, or you have to manipulate the situation to make that happen?
In Myxococcus xanthus, it's relatively easy, but that was the reason why that organism was chosen. Other myxobacterial species also make fruiting bodies, but they tend to be a lot more finicky. Stigmatella rantiaga, Syringium cellulosum. Some myxobacteria, like Enteromyxobacter, the halogenans, actually doesn't seem to make fruiting bodies. But Myxococcus xanthus was chosen because it makes them pretty reliable.
Does Myxo express or rather, does it excrete strong biosurfactants, and does it ever produce lots of extracellular matrix like other species? Does that play a role in its mechanical properties and formation of fruiting bodies?
So like a normal biofilm, it sort of exists in its own self-generated matrix. Exactly what constitutes it is still somewhat unknown. It makes this stuff called slime. This is something that biologists get to do, where we don't know what it is, so we describe what it, or we name it according to what it looks like. The kind, exactly what slime is made up of, we don't know. There is actually a hypothesis that it's part of the propulsion system, that the extrusion of slime is actually what drives the thing forward. Other people don't believe in that. Some people that just think it's a form of stigmergy, where essentially one cell lays down a slime trail and then the next one follows behind it.
And does that include some surfactants as well, do you think?
I would assume so, but I don't know.
Okay. Could you explain more why you expect some analogy with oswald ripening? Because I don't see at all the connection.
Okay, this is probably why Oleg told me not to mention it. So what I'd like to do is talk with you and that guy, and then maybe the two of you can get to fighting, and I don't have to sound stupid. So
I don't need to fight. I'm just trying to understand, but I don't understand.
Well, I'm not sure I do either. So how about the three? Well, I'll buy you guys a
sure.
Yeah, so I think it's not real the orthogonal, but I think it's basically a combination of, in the same conditions, the same cells are doing two different things. We have two subphenotypes. One of them are spreaded, the other one are aggregated. Basically, we have some cells spread, and they decide to aggregate. When they aggregate, some of them die, so the rest get as a food, they keep on spreading, they get another side. So I think the mechanism, the internal mechanism is quite different from the statistical mechanics of the organism. So maybe at some abstract level equation might be similar, but we couldn't figure that out.
I have a couple of questions about just the movies. It wasn't so clear to me what I was seeing. I saw these like gray kind of uniform things, and then I saw these black things forming. And can you just explain a little bit what I was seeing there? I mean, I was seeing the individual cells, does that make sense?
So you know, the self-generated matrix that I was talking about sort of makes it so that the individual cells are obscured. So the swarm appears as sort of one entity, even though it is composed of millions of cells. So I apologize for not making that clear. That's one of those, when I give a talk to microbiologists, I leave out stuff, and I shouldn't have. So the movies that you're referring to were made with straight up normal bright field microscopy, which basically means that there is some relationship, it's assumed to be somewhat linear, between the darkness and the density of what the light is shining through. So you can assume that the grayer areas are more cells piled up on top of each other in that region, and the lighter areas are fewer cells. So you're looking essentially at density, and then the aggregates, the black circles that you were looking at, are the fruiting bodies that are sort of growing, so they've now essentially reached the threshold of black and are now sort of completely black, even though they have a dome shape. You can't sort of see what the shape is based on the grayscale.
So are you assuming that they're hemispherical? Because I would guess the model must make some kind of prediction about how they exist in the third dimension, but your images don't allow you to deduce that. So then when you're counting, like, whether or not, when you were doing all these statistics on the things disappearing and growing, another possibility is they could just be changing their height in the third dimension, but there should be definite predictions from the model about what's going on, and have to check those.
So that's actually a couple of good points. Let me sort of handle them one at a time. So actually, one of the first guys that I ever collaborated with was a guy named Adam Arkin, who was at Berkeley, and he was helping me model some stuff. And I remember getting a question very much like that about the third dimension, sort of stumbling on it, and he interrupted me and just said, look, the pattern's two-dimensional, so we can model it in 2 dimensions, and it's still something worth studying, even if we don't see the third dimension. So we're actually sort of modeling the third dimension separately, but it adds a level of complexity which I don't think is a bad thing to treat separately in terms of image analysis. You're right, in terms of modeling, once they get there, they simply can't stop. One of the other guys that does modeling, a guy named Mark Albert at Notre Dame, he models Myxococcus xanthus. That's essentially what happens, is that fruiting bodies are just places where cells stop. And absolutely, if it's making a defined shape, it's doing something. They're doing something to make that shape, but we haven't gotten there yet.
And then just finally in the images, at certain points I saw like these waves of black and white appearing in the gray, and they would appear and disappear. So what's going on with that? What sets the wavelength of those? And
I would say it's a very good question. Why don't we discuss it over the coffee, because we are running a little bit late. But I just know it's a subject of separate talk, the way it makes it represents. So let's thank Roy and all the speakers for coffee is here now.
Stop recording.